hydrogen bonding too. So fragmentation is like a small piece of the moiety, small piece of chemical. It can bind into the enzyme. Then we can use that small piece to elaborating drug design. Okay, so from small piece to make a bigger. That's a fragmentation drug design. Okay, so we do this whole activity. Then we're gonna end up with about 100 chemicals. Okay, about 100. Then everybody sit down and then looking at the 100 structures. Okay, and that 100 structure would be a gate to the computer assisted drug design, which is the X-ray. Okay, so these people taking 100 compound, put it into the enzyme, and then take the picture of the X-ray. So we can tell that from the picture how this chemical inside of enzyme or receptor. Very good, right? This is the modern science. So what they do is they're taking the enzyme and the chemical, okay, and put inside of the enzyme. Usually enzyme is a very good one because of nice structures. And then make the crystal. And just like you go to hospital, take the X-ray, put in the X-ray beam. See how that arrangement of atom inside of the structures. And then do the computer calculation, they give you nice pictures. How this guy is sitting inside of the territory. From those pictures, and also the structure-activity relationship is another technique. You modify the part, small part of the chemical to make the better, not only the well binding. I mean, they don't understand the binding, right? Okay, how love each other's, okay, in the enzyme or receptors. And then another very important part is Noel is the expert about how well behave in the human body, toxicity as the pharmacokinetic, how well inside the body and get out. Those kind of property can be controlled by this structure-activity relationship. So all this thing, we're working in the chemistry department, the biology have to make in vivo assay. The in vivo assay means animals models. Okay, so like most of the case, genetically modified rat is a good model. So they make the rat and modify the genetic, okay? Their gene was modified, so certain feature is changed. It's a very expensive rat actually. You can make it in this one in vitro, in vivo assay. There's a lot of people selling rat, making fortune. Also the academia involved with that too. The genetically modified rat is very, very expensive. Like one rat is about, I don't know how much is it,
Over $1,000.
Yeah, it's around $1,000, sometimes 5,000, one rat. So making rat is a very profit job. Anyhow, so in vivo assay, we taking this kind of research, the chemical, we making chemical, and they are developing vivo assay, and then we combine these two, see whether this chemical really working in the disease. Okay. So all this activity boils down to identifying pharmacophore. Pharmacophore is a chemical moiety which is we know how interact with our target. Okay? So this has become drug. Now, this is about this one, from here to here, we're taking about 5 years or 6 years. So this diagram is kind of like, give us how this launch product is originated. Okay? So as you see that, about 75% of the compound is using this technique, hydro screen, chemical department, SARs, and getting the pharmacophore and optimizing as much as you could. Okay, so this is like the evidence how most of drug company making drug. Okay, about the 16% is from biological. That means you're currently, there's like a new trend evolving. It's a biological drug because the biological drug is originally in your body. In most of the cases, protein. Okay, and then protein is using as a drug right now. So that's kind of like a biological area. Normally, they making new amoeba to make this protein. So then the preclinical area. Before you go to clinical, you have to go into the different animals. See whether this drug is well behaved inside of the biological system. So normally, you go from rat, dog, monkey, okay, to see this drug has blood levels enough to cover up the potency or efficacy of this drug. Okay, so you checked all the animal species to see whether it have enough behaviors. Okay, and then also during that time you're going to see about toxicity too. Every chemical has a toxicity. No chemical is safe, right? Even if you take the aspirin of a kilogram, you die. Okay. So chemical is always danger there. At this moment, they are working on the patents. Okay? Just before you go into the FDA, they're working on the patent. So they can have extreme long time patents, 20 years. Okay? Because as I talk about the clinical trial, you need about 5 to 6 years. So you're losing 5 years right there without making any money, just spending money for 5 years. So they file the patent at this moment. Okay. And then also that the metabolism is, it's kind of like a metabolism, and then the pharmacokinetic kind of data is like a black box. Unless you put it in and the monitoring, you don't know. You cannot really predict how good it is. And so we have to keep on getting the drug metabolism data. How it's gonna, cleave inside the body. Most of the drug will be about the cleave in your liver very fast. So your liver is very good. Make yourself very safe. So, and then the toxicities are very important. You don't want to kill the people for making the treatment, right? So make sure that's safe. And then in this time, they designed the manufacturing process. Initial target is about 10 kilograms of drug. 10 kilograms is about the same size as a rice bag, maybe, right? But to do that is very, very difficult. You have to make sure 99.5% pure, 10 grams. Okay, 99.5 is very, very difficult numbers. You can make easily 90% pure material very easily. But when you go 99.5 or 99.95, your hair is gonna slip up. Just the dust can interact, right? So this process is very, very difficult. And then we're gonna go clinical trial. The first thing you have to go is take all this information about animal study, drug efficacy study, and so on, give to the FDA, investigational new drug applications. You file to the FDA, and then you wait for 30 days. If FDA say nothing, then you can start. But normally they say something. I need another papers. Why don't you do this test? Okay? So you give all previous experience, chemical structure, and then toxicity in the animal, what kind of stuff have to give to the FDA. Now once you get that FDA clear from your blood, then you're gonna go clinical study. So this is the brief summary of the clinical study. Phase 1, we want to address the question about safety. How safety this drug is, or chemical moiety is. Phase 2, we're going to talk about efficacy. How effective this drug for the patients. And then phase 3 is a long-term efficacy for the patients with the more variety of patient groups. Okay? And then you're going to have a new drug application. From this moment, you're going to making money. So far, you're just spending money all the time. From this, you're going to make the TV advertisement and so on, okay? Like in a 2-bathroom for Cialis. That's very, I don't like that commercial. And then after that, phase 4 is kind of continuously they monitor how the drug is behavior in the market. Okay, then go clinical study 1, phase 1. It's about a very small group, 20 to 80 group, but they have to be a very healthy person, doesn't have a disease, okay? And then they give you some evaluating safety, so they're gonna give you like, small amount first, middle amount more, and more, and more, until they say, I'm so dizzy. Okay? I'm so dizzy, I got vomiting, I hate this song. Okay? Then they stop. Okay? So it's a very small amount. And then see, it is give you hair loss, or it has a uncomfortable stomachs, or their heart is pumping too much, kind of like observe everything. And to that also, they check the blood level. How much you put it in, 1 gram of the drug in the people, how much in the blood. Why? Because most of drug is taken from oral, right? You take the pill. And it doesn't, if you take a pill, have to go into your digestion system, get into the body and get inside the blood. So everything like 1 gram you take it in doesn't mean that 1 gram in your blood, right? So make sure the blood level is right to cover up the disease area. And this is about